Okay, I think we just start broadcasting. So good morning or good afternoon, everyone. This is a great pleasure for me to introduce my dear friend, Professor Peter Vandenabeele, one of the guardians of the cell death secrets and a caretaker of honeybees. We were just talking about that. And finally, a professional bowling player. So Peter obtained his PhD at the Ghent University in the Laboratory of Molecular Biology, heading by Professor Walter Fiers. In 1996, he joined the VIB Ghent Center for Inflammation Research, everything Belgium, of course, and where he became the head of the cell death and inflammation unit. His research activity focused on multiple molecular mechanisms of all sorts of cell death modalities, including the ones yet to be discovered. Peter has devoted himself to mentoring young scientists, and many of them has become PIs. Actually, he has been the founder and the director of the bachelor and master program in biochemistry and biotechnology in the Faculty of Science at the Ghent University. Well, there's a long list of awards that Peter received, members of the Board of Governors, member of the Royal Flemish Academy of Belgium. Anyways, okay, I will cut it short because we don't have much time. Thank you so much again. Peter has been participating in this course since the beginning, okay?
Yeah.
Thank you, Peter.
Okay. Thanks for the invitation, and it's my pleasure to give this talk. It's always a challenge to come to something comprehensible, so I hope I will succeed this time. So I will talk about necroptosis, and first of all, a nice view on Ghent. Every evening, the sky is like this in Ghent, and during the day, we have a blue sky always, no rain. And then our institute, you can see here the same blue sky, and this is a view on our campus with the several buildings of the Flanders Biotechnology Institute. So it's a nice area. I would have preferred to have my lab here in the castle, but we are in a more modern building. So I will talk about necrotic cell death, and this is an artistic interpretation of one of the stepfathers of one of the PhD students. I like the friendly way of representing necrotic cell death. So I will talk on necroptosis, the history, how it originated from TNF research, then a bit on the molecular mechanisms, and then very short on the therapeutic potential. I will also talk a little bit about the evolution of the necroptotic pathways. So after me, Jonathan will give his talk on the ZBP1 and make more the connection between cell death, including necroptosis and antiviral activity. So let's start with the history of TNF research. It's always good to go back to see where the fruits that we are harvesting now, to go back in history and how things started. At the moment, we have more than 200,000 articles on TNF. If you compare that, for example, with another topic like P53, it's 150,000, 15,000, and caspase is like 130,000. Now, it all started TNF research in 1890s. And there was William Colley who treated patients with extracts from bacteria, the so-called Colley toxins. And those extracts were prepared from skin infections. And those extracts contain Streptococcus pyogenes and also Serratia marcescens. In fact, it was already based on observations by a German physician in 1888 that sometimes skin tumors could disappear in patients with skin infections. So then Colley continued on that and used bacterial extracts from those skin infections. And then about 50 years later, it was identified that LPS from those bacterial extracts was in fact the compound that was inducing a tumor regression. So then we had to wait again another 10 years to demonstrate that indeed injecting LPS in, for example, tumor models in mice was inducing a tumor regression. And then a very important contribution was that LPS was shown not to work directly in its anti-tumor response, but that LPS was inducing in the animal a factor that was responsible for the tumor regression. And from then on, it was called tumor necrosis factor. So in fact, has been purified by injecting hundreds of rabbits with LPS and then purify the factor by testing it on certain tumor cells and see its tumor reducing activity. And it was in 75 that Lyot Old identified this factor and got the sequence of it, tumor necrosis factor that was induced by endotoxin. And he was able then to provide it in tumor models and induce necrosis of tumor cells. Now, the importance of this observation is, of course, that it, for the first time, it made the link between bacterial infection and the production of endogenous factors that are able to induce tumor regression. In that sense, William Colley can be considered as the godfather of immunotherapy. Now, the next time period is, of course, the period of cloning, and my mentor, Walter Fiers, who died several years ago in 2019, did with his group the cloning of TNF. At that time, I was starting a PhD student. And people expected a lot from TNF as an anti-cancer cytokine. But on the other hand, TNF was also a central mediator of inflammation. So it had, in fact, two phases. On the one hand, it was a potent way to induce cell death in certain tumor cells. On the other hand, it had also potent pro-inflammatory properties. So this led in 96 to the application of TNF in patients, and that was done by isolated lymph perfusion for sarcomas on the leg. And of course, because of the toxicity of TNF for inducing systemic inflammatory response syndrome, the circulation had to be separated from the rest of the body. Therefore, the name isolated lymph perfusion. And in that way, sarcomas could be very efficiently treated, especially in combination with certain chemotherapeutics and also with combination of interferons. But anyway, as you can see, this had only limited applications. And at the moment, TNF is not really used anymore directly for anti-tumor therapy. Although, as you will see at the end of my talk, there is a revival of the use of TNF in considering TNF in anti-tumor therapy. Then the next, let's say, failing of TNF was the identification that TNF is the same like cachectin. And cachectin, as the name says, is a factor that is inducing extreme wasting of patients during sepsis. And it was Bruce Beutler and Anthony Serrami that identified that TNF was in fact identical as cahectin. So the idea came up to block TNF in the conditions of sepsis in sepsis patients. However, also this clinical trial failed because, as you all know, TNF is also needed for our protection against infections. And especially in sepsis patients, blocking TNF did not give the best outcome. But the main, let's say, success of TNF was, of course, the aspect of TNF that it is inducing, that it is involved in many inflammatory diseases. And one of the first double-blind randomized studies of blocking TNF in rheumatoid arthritis in 94 was published in the Lancet, and that leads to the application of blockers of TNF in many inflammatory diseases. And although treatment of anti-TNF in sepsis failed, and also using TNF in cancer failed, the treatment of inflammatory diseases like rheumatoid arthritis, inflammatory bowel disease, and psoriasis has been a real success. And at the moment, it's different anti-TNF strategies have been developed by different companies, and you know for sure the names like infliximab, adalimumab, Humicate, etanercept, and those are anti-TNF biologicals that are one of the, well, which is the biggest market for biologicals at the moment. There was a market of $40 billion in 2017, and now it's already increased to $180 billion a year. Now, TNF has many activities on many different cell types like T cells, hepatocyte, fibroblast, myocytes, endothelial cells. And all those TNF activities are mainly associated with the capacity of TNF to activate NF-kappa B. But on some limited cell lines, TNF can also induce cell death. And of course, we got very much interested what is in fact the decision point between TNF inducing in certain cells only gene activation, and what is the decision point of TNF inducing also cell death in some cell lines. And then with the discovery, let's say in the mid-90s, of the apoptotic pathways, and we have been discussing that yesterday with Seamus Martin, the extrinsic pathway, starting from the receptor recruitment and activation of caspase 8, and then either directly going to the executioner caspases or requiring the intrinsic pathway through the release of cytochrome C, leading to APAF1 activation, the apoptosome, and caspase 9 activation. This communication between the extrinsic pathway and the intrinsic pathway, let's say, was elaborated in the mid-90s with many different contributors, as shown here. Now, at that time, I was doing my postdoc, and we found a very strange observation. And it was also mentioned yesterday by Richard Luxon, that sometimes you find things that do not fit in all existing models. So what we found is that CRAM-A, when you expressed it in cells, that instead of blocking cell death, we got 100-fold to 1,000-fold sensitization of cell death. So that was an observation that went against all current thoughts about apoptosis, because CRAM-A is targeting caspase 8. So we had here the remarkable paradoxical observation that cells expressing CRAM-A were even more sensitive to TNF induction. And this TNF induction led to the production of reactive oxygen species. And at that moment, we could make easy models, as you can see. We were thinking that CREM-A was blocking some caspase that was involved in the protection of mitochondria. And if you block this caspase, then you get increased mitochondrial ROS production that then was leading to necroptosis. So you can see 20 years ago, the world was very easy, very simple schemes. You will discover that everything became much more complex nowadays. Now, the fact that TNF is able to induce two different types of cell death was, in fact, in a retrospective way, already discovered. It was Scott Laster that found that on certain cell types, TNF will induce blabbing and apoptotic cell death, while in other cell lines, it will induce swelling of the cell and explosion of the cells and more like necrotic cell deaths. And so we were able to repeat that in the context of one particular cell line. And when we stimulated cells, for example, here, fibrosarcoma, L929 cells, when you stimulate them with antifas, you can clearly see induction of apoptosis with blabbing, also internucleosomal cleavage of DNA, and then later on secondary necrosis and staining of the plasma membrane permeabilization and staining of the nucleus with propidium iodide. While in the case of stimulation with TNF, we got necrotic cell death that, as you can see, the nucleus remains intact and is associated with swelling of the cells, and there is no internucleosomal cleavage of the DNA. So it was clearly established from then on that in the same cellular context, you can induce two different types of cell death. And we were also able to repeat this fundamental observation also in vivo. And for example, here you see injection of TNF in mice, and it is inducing a systemic inflammatory response syndrome. When you co-inject with ZVAD, which is blocking caspases, we got an enormous sensitization of the lethality of TNF injection, and the mice were dying immediately. So that was indicating that this necrotic form of cell death may also be operating in vivo, and was in fact the first demonstration between necrotic cell death and the inflammatory response by systemic inflammatory response syndrome. So this connection has also been described yesterday by Seamus Martin. So then so this is in fact what we observed, that in a model of TNF-induced systemic inflammatory response syndrome, that when you block caspases, you get an enormous sensitization, much higher inflammation and cell death, leading to sensitization of the lethality. So from that moment on, my world became dichotomic. So I was only thinking about either apoptosis or necroptosis. There was also a very important contribution of Jörg Chop in 2000, who demonstrated that RIPK1 was the effector molecule in a cell death process that was different from apoptosis. Also from the group of Jörg Chop, who died unfortunately, there was also the concept growing that TNF can induce two types of complex. So there is complex 1 that is responsible and that is associated with the TNF receptor 1, and that is responsible for NF-kappa B activation and survival of the cells. And one of the genes that are induced are, for example, BCL2 and also FLIP caspase inhibitors. And then there is also the complex 2, which is cytosolic, and this cytosolic complex is important for the induction of cell death by apoptosis. So this concept of complex 1 and complex 2 will come back later during my talk. So then we come to the period of 2009, 2012, and then the necrotic cell death pathway induced by TNF has been understood in molecular terms. And a real important contribution in that was the identification of a compound, necrostatin, by Alexei Dechterev and Yunying Yuan, that was able to block necrotic cell death induced by TNF. And later on, contribution by Xiaodong Wang, also Andrew Oberst, Doug Green, Bill Kaiser, and Han identified RIPK3 and RIPK1 as important mediators of necrotic cell death, and that the kinase activity is important for that. And very interesting, apparently the explosion is not a passive phenomenon. There was also a pore-forming molecule identified by Liming Sun in the group of Xiaodong Wang. And further, also the structure has been elaborated by Murphy and Xabotar. And that was involving MLKL as the executioner of necroptosis. Then later on, it was found that RIPK1 has also two phases. So RIPK1 as a scaffold is very important for the inflammatory, direct inflammatory signaling and for the gene activation, while the kinase activity is important for the cell death induction by apoptosis or by necroptosis. And this decision between, let's say, survival and inflammatory gene activation signaling and cell death signaling involved several checkpoints that have been identified here in Ghent by Mathieu Bertrand, Geert van Loo, but also contributions of Henning Walksack, Doug Green, John Silk, and Pascal Meyer, and also Manolis Pasparakis. So we are now at the level of RIPK1. I mentioned you that RIPK1 as a scaffold can induce cell survival and gene activation, and that the kinase activity is important for cell death induction. Now, how is this decision between, let's say, survival function and gene activation versus cell death induction regulated? So what are the checkpoints that control this decision? We will see later that there is also another checkpoint decision between the decision to die by apoptosis or to die by necroptosis. So let's have a look at RIPK1 at the cross point of survival signaling and cell death signaling. Now, RIPK1 belongs to the family of RIP kinases, and there are two important RIP kinases, RIP kinase 1 and 3, that are implicated in cell death pathways. Now, RIPK1 and RIPK3, they have a special domain, and this is called the RIM domain. And this RIM domain allows, in fact, aggregation of this RIP kinases. There is one other gene, ZBP1 and TRIF, two other genes that also contain this RIM domain. So in fact, in vivo, in mammals, we only have, let's say, 4 proteins with RIM domains, and they are all implicated in necroptotic cell death. So then RIP3 was identified in the necroptotic pathway, and also in viral pathways, and Jonathan will talk more about this. Now, the RIM domain is very important because the RIM domain can form antiparallel beta sheets that allow aggregation of factors that contain the RIM domain. I mentioned you already TRIF, ZBP1, RIP1, the 4 genes that contain this RIM domain, allowing aggregation. There are also many viral proteins that also contain this RIM domain that will interfere with our cellular RIM domains to block the cell death that is elicited during viral infection. But Jonathan will surely tell more about this. So when we look at the necroptotic pathway, so it can be induced by TNF receptor family members, then it involves RIPK1 kinase activity, and this RIPK1 kinase activity will then further activate RIPK3, that then will phosphorylate MLKL, and that leads to the formation of the so-called necrosome complex. It can also be induced from toll-like receptor family members like toll-like receptor 3 and 4, and then it involves TRIF adapter, which can directly interact with its RIM domain with RIPK3 and lead to the activation and MLKL phosphorylation. And then it can also be induced, and Jonathan will tell more about this, by ZBP1 that can directly activate RIPK3 and induce phosphorylation of MLKL. I will skip this slide. So I mentioned you that we have RIPK1 as a survival factor, and RIPK1, its kinase activity involved in cell death. Now, how is this RIPK1 regulated? So it has many different checkpoints. And RIPK1 is a universal factor that in most cells is absolutely required for survival. So people have made RIPK1 knockouts in many different cell types in the skin, the intestine, the lung, the kidney, the brain, tumor cells, immune cells, the heart. And from the moment that those specific cells lack RIPK1, they will spontaneously go into apoptosis. So that means RIPK1 is absolutely required for the survival of many different cell types. So that means also that the cytotoxic potential of RIPK1 as a kinase is heavily controlled by different checkpoints. So RIPK1 can, on the other hand, its kinase activity has been shown to be activated in many different conditions of cellular stress. This can be metabolic stress, this can be mitochondrial stress, this can be TNF receptor signaling, this can be viral and bacterial infection, genotoxic stress. They can all result in the activation of RIPK1, its kinase activity, and then induce cell death, mainly by apoptosis. Now, how is RIPK1 controlled? I mentioned you already the checkpoints that, in the case of TNF, keep RIPK1 in the survival modus. And when those checkpoints are inhibited, then the system can turn into a cell death modus, either by forming a cytosolic complex 2 that can lead to RIPK1 dependent apoptosis, or can lead also to RIPK1 independent apoptosis. On the other hand, as I've shown with the Cremé experiments, when you block caspase 8, RIPK1 can then induce a necroptotic cell death. So when we look, so complex 1 is, in the case of TNF, will induce survival signaling. This is kept in check by many different checkpoints, and those checkpoints are ubiquitination, linear ubiquitination by Lubac, or K63 ubiquitination by the SIAPs. But also phosphorylation events keep RIPK1 in a survival modus. For example, IKKs from the NF-κB pathway can directly phosphorylate RIPK1 and keep that in a survival modus. Also, MK2 and TBK kinases can phosphorylate RIPK1 on certain residues and keep it in a survival modus. However, when we block, for example, those ubiquitinating enzymes, or we block those kinases or deplete the genes of it, then the system turns in towards cell death and can be a complex 2 formation that is inducing either RIPK1 independent cell death, RIPK1 kinase dependent cell death, or in the case that caspase 8 is blocked, it can induce necroptosis. Yeah. So here you can, for example, see if it works, but it doesn't work. You could see in the case of cell death induction that you have RIPK1 complex formation. And when you use RIPK1 kinase inhibitors, this complex formation is completely inhibited. To conclude this part, many different types of physiological stress or even homeostasis conditions can, stress conditions like infection, injury, metabolic stress, can impinge on the TNF signaling. And then TNF can either lead to survival, because it's a factor that is required to combat, for example, infection, or can induce systemic inflammatory response syndrome and cell death and lethality and autoimmunity. On the cellular level, we have another checkpoint and also dichotomy between RIPK1 as a survival factor and RIPK1 as a cell death inducing factor. And then a next checkpoint is at the level of cell death. It can be either apoptosis or necroptosis, depending on the levels of caspase 8. And even in the decision of necroptosis, we have again a dichotomy and a kind of checkpoint, because there are MLKL protective mechanisms. Once the pore is formed, this pore formation can still be counteracted, for example, by escort mechanisms or by degradation in the endosomal lysosomal pathway. So you see that this combination of all those dichotomic decisions and the presence of all those checkpoints that can come to opposing decision will lead to a very complex network. And in fact, this kind of opposing decisions is very important because it allows redundancy and fine-tuning of the system. It also allows that in a population of cells, individual cells can come to different decisions. And I think that those different decisions of individual cells in a population is very important for moderating the response and also to come to an adaptive response in the system. And I think that the fact that we have this kind of mixed responses in a cell population avoids that the system will crash. And in fact, it allows us to survive in stressed and diseased conditions. Otherwise, we would die immediately. But we can live with quite some stress and disease conditions. And it's in fact a very important factor for resilience. So in fact, it reminds me a little bit of how Belgium looks like. So Belgium is a very small, complex country. It's half of the population of Sao Paulo, but it has a very complex governance system. So we have about 11 million people. There is one federal level. There are 3 regions. There are 3 communities: Flanders, Wallonia, Brussels. We speak Flemish, we speak French, we speak German, and there are 7 governments. And thanks to this complexity, there are several dichotomy and checkpoints in the system. And thanks to this complex situation, which resembles a lot TNF signaling, and that's also why Belgians can understand complexity in molecular biology and signaling, is that we survived, for example, Belgium as one of the best countries, the financial crisis of 2010, 2011, with almost 600 days without any government. So the system is very redundant and resilient. So complexity and imperfectionism allows resilience. And I think this is interesting thoughts for times of extremism. But I would like to continue another 5 minutes. Is this possible? Okay. So there are many ways to die. We have different pore-forming mechanisms, and there are different ways how you can permeabilize membranes. There is the carpet model, in which amphoteric proteins or peptides will interact with the phospholipids and can induce instability. Or you have the barrel stave models, where you have very nice protein consisting pores, or you have a kind of mixed model, the toroidal model, in which proteins and phospholipids form, with positive bending, a kind of mixed lipid protein pore. Now, we have seen that in cell death there are many different types of cell death pore induction. In necroptosis, it is MLKL. We have seen that in apoptosis, Bax-Bak play pore formation at the mitochondrial level. If apoptosis can also continue in late apoptosis or secondary necrosis, and has then also a permeabilizing mechanism by activating gasdermin E. Then we have pyroptosis, in which directly gasdermin D is activated and leads to the release of IL-1 beta, or pyroptosis can also directly activate by caspase-3 gasdermin E. And then we have also ferroptosis, in which oxidized lipids can lead to plasma membrane permeabilization. And even when all those pores have caused plasma membrane permeabilization, we have on top of that another mechanism, NingJ1, that is sensing the swelling of the cell. And this NingJ1 will then cause a final membrane rupture and will induce the release of large DAMPs. So necrotic cell death is really about making pores into the plasma membrane. It has to do with the essence of life, is keeping your plasma membranes intact, and necrotic cell death has very active mechanisms to interfere in that. And how is MLKL working? So I mentioned you already that it was first identified by Liming Sun in the group of Xiaodong Wang. And MLKL is activated by RIPK3, is then recruited to the plasma membrane and goes interaction with phosphatidyl inositol phosphates, will oligomerize. And I mentioned you that even at that level, there can be a detoxification mechanism. For example, escort mechanisms can lead to the formation of microvesicles that lead to the release of the toxic MLKL, or the MLKL can be internalized and then form the so-called intracellular multivesicular bodies that then can lead to extracellular vesicles or lysosomes. So I think it's very important to see that there are continuous kind of detoxifying mechanisms to allow again the restoration of the plasma membrane integrity. And how is MLKL working? In fact, it's still not known. So when you look, the toxic part of MLKL is in the N-terminus and contains 4 helical bundle domains. It has been nicely described by Ana Garcia Saiz. And those are amphopathic alpha helices with let's say, lipophilic residues at one side, and then the the hydrophilic residues on one side and the hydrophobic ones on the other side. And this is somehow interacting with the membrane and is causing probably a kind of destabilization. It's not clear how MLKL works, whether it's working like a carpet mechanism or a toroidal model is still to be discovered. Now, quickly, some evolutionary aspects of necroptosis. You could think necroptosis is very important, but when you look into the presence of the RIM domain containing proteins like ZBP1, RIPK1, and RIPK3 and TRIF, if you look, they have been developed only rather late together with the development of the adaptive immune system. And interesting enough, there are some organisms like birds that completely lack ZBP1 and also lack RIPK3. So in fact, if you eat chicken, this is ZBP1 and RIPK3 free. So this is safe for necroptosis. The same is true for the placentalia or the marsupialia that do not contain ZBP1. They don't contain RIPK3 and MLKL. So those animals, like the Tasmanian devil and opossum, and the wallaby, they are completely free of necroptosis. Also, the carnivora, for example, they lack, like dogs and cats, they lack MLKL. So if you eat dogs and cats, no problem with necroptosis. So it is, of course, quite interesting to think why this kind of necroptotic pathways have been lost in birds, in carnivora, and also in marsupialia. So what has been the driving force for this loss? We can discuss that. And then, of course, we come to the last point, is the targeting of RIPK1. I can be very brief in that because that will be elaborated in much more detail by Domagoj Vucic in one of the later talks. But there have been developed RIPK1 inhibitors, and the prototype is necrostatin 1. And the whole idea is by blocking necroptosis that you can block the inflammatory cascade that is initiated by RIPK1 dependent cell death, either by apoptosis or necroptosis. And in fact, the principle was shown by us in 2011 that in a model of systemic inflammatory response syndrome with mouse TNF, we could block with necrostatin the induce complete survival, and also RIPK3 knockout mice, they did not die from a systemic inflammatory response syndrome. Now, there have been many diseases identified in RIPK1 could be targeted. I will not go into detail. The Domagoj will talk more about this. Also, RIPK3 looks like a very interesting target. There are diseases in cancer, but also degenerative diseases, also viral infection, in which necroptosis is really important. Now, targeting necroptosis could be an alternative for the anti-TNF strategy because TNF is required for the antibacterial and anti-infection responses. So by targeting specifically the kinase activity, we may still keep the anti-infection responses, which are mainly dependent on the scaffold of RIPK3, and blocking the cell death response could save us from their contribution to inflammatory diseases through the induction of cell death. So at the moment, RIP1 inhibitors are tested clinical trials in IBD, also in psoriasis, and also in rheumatoid arthritis. But you will hear more by Domagoj about this aspect. And then I come to my last slides. So we have really experienced since 2000 an explosion of different cell death modalities. especially necrotic cell death modalities. The most important cell death, as Seamus mentioned yesterday, is of course apoptosis, because it's also through efferocytosis involved in resilience, in regeneration and resolution of inflammation. So sometimes I think we live in a period of necrotic hallucinations. There are so many necrotic cell death modalities described. And I think, like you have chat GPT hallucination, I think we also have now necrotic hallucination. But if we want to say something on those different cell death modalities, we can say apoptosis is for sure the main cell death modality during homeostasis, social context, development, infection, injury, metabolic stress. But on top of that, during infection, especially pyroptosis and necroptosis are very important. And then you have many, let's say, cell death modalities, which I would call are more a consequence of so-called cell death by sabotage. If certain systems do not operate well, like redox systems, like metabolism, like autophagy, their absence of those protective mechanisms may induce so-called sabotage in the cell that will lead to other cell death modalities. But at the end, cell death is important because the way, whatever apoptosis we are dying, we can, by the way that we are dying, what kind of pores we use, we communicate with the environment. And communication between cell death and the immune system and other cells in our body is extremely important for during infection, degeneration, injury, inflammation, efferocytosis, resolution, and regeneration. And then I come to my final slide. I'm so grateful that I have fantastic people around me that do keep me in check. So they are my important checkpoints. And thanks to them, I'm able to tell you nice stories. So I'm ready to take questions.
Super, thanks, Peter. It was great as usual. I would ask Igor and Leticia to open the mic and ask their questions. Any other questions we will leave to the end, and then combine questions for Jonathan and
that would be good.
and Peter. Okay, but Igor, please go ahead. You first, and then Leticia. Can you ask your questions?
Hi, thanks for the lecture, Professor. And I have a question. In case of autoimmune disease, can massive cellular destruction by the immune system be considered something close to necrosis in relation to the damage caused? An example as psoriasis.
Yeah, so it has been shown that in the skin, especially in the skin, that the cell that is that you have a kind of mixture of cell death. Of course, you have apoptotic cell death, but maybe also Jonathan will talk about that. But especially the skin is very sensitive to ZBP1 mediated cell death. And there we have, especially because there is also interferons release, and that leads to upregulation of ZBP1. And there we have an important contribution also of necrotic cell death. So I think by probably by blocking strategies of RIPK1 kinase activity, and probably also in combination of controlling ZBP1 induced cell death, we can surely contribute to the resolution of psoriasis-induced inflammation.
Thank you, Professor.
Thank you.
Hi, Professor. Thanks for the lecture. It was actually very interesting.
Thank you.
And my question is about TNF is used nowadays to treat some disease more specifically? And what is the mechanism that TNF acts to treat
Yeah.
or to resolve the problem?
Yeah. So in most treatments, as I indicated, is in fact by blocking TNF, because TNF plays an important role in all kind of inflammatory conditions. Nowadays, because now we understand much more about the checkpoints, people are going back to the original activity that was discovered with TNF, like the name says, tumor necrosis factor. And by blocking certain checkpoints, we can sensitize the cell death induction of TNF in many different tumors, and also in tumor-associated endothelial cells. The problem there will be, of course, the systemic toxic effects of TNF to keep that under control. And therefore, I think a combination with strategies where you have targeting more specifically to tumor environment, your TNF or the sensitizing factors may open new opportunities, and people are exploring those of, let's say, bringing back TNF in the anti-tumor treatments.
Thanks,
That's
Professor.
great. That's great. I think we have time for another one. Lucas, would you ask your question, please?
Hey, Peter, thank you for your presentation. It was
Yeah.
beautiful.
Thank you.
My question was, can I rule out necroptosis if I have one alpha helical amphipathic peptide that activate caspase-8? It was one way to
Yeah.
ask what's the relationship between caspase-8 and necroptosis?
Yeah, so yeah, I didn't go into the details. So caspase-8 is negatively controlling necroptosis. So, and it has been recently discovered by Kim Newton and also the people from John Silk, that caspase-8 is in fact cleaving RIPK1. And RIPK1, if it is not cleaved, it will accumulate, and higher levels of RIPK1 will be more prone, and in the absence of caspase-8, will then better initiate necroptotic cell death. So in fact, it's a question of blocking caspase-8 activity so that it cannot contribute to apoptosis anymore, but on the other hand, increasing the levels of RIPK1, and those increased levels will then initiate the necroptotic pathway. And that's what people have found, that caspase-8 knockout mice, they die spontaneously by necroptosis. So in fact, we continuously need this checkpoint of caspase-8 to control, even in homeostatic conditions, TNF toxicity.
Thank you.
Excellent. Okay, so last question from Rafa, and then we will move to Jonathan's presentation.
My question is about the moleculars. It's capable to permeabilization of the membrane. I was thinking about the part of the presentation, and I see the gasdermin D
Yeah,
is related to late apoptosis and paroptosis. Could
indeed,
you
yes.
talk about a little more about the same molecule being related to the two
Yeah.
types of cell
Yeah.
death?
Gasdermin E, not
Yeah,
D.
gasdermin E. Sorry.
Yeah. So gasdermin D is associated with inflammasome activation. You will hear more about that because gasdermin D is activated by caspase-1 and caspase-4 and 11, while gasdermin E is activated by caspase-3, the executional caspase of apoptosis. So in conditions where there is no uptake of apoptotic dying cells, for example, overwhelming apoptosis, the cells can continue and will then lead at the end to the activation of gasdermin E, and lead then, as we discussed yesterday, to secondary necrosis or late apoptosis. In some cell lines, caspase-3 can be directly activating gasduimin E. In that case, I would talk about pyroptosis, because you do not develop the apoptotic features like blabbing. The cell will directly activate the pore-forming mechanism and lead to explosion of the cells by the pore-forming mechanism.
Thank you so much.
All right. Thank you, Peter, so much. We will come back to novel questions after Jonathan's talk.
Okay.
All right. So
Okay.
with
Thank
that,
you, Peter.
with that, I would like to thank very much Dr. Jonathan Maelfait for accepting our invitation. Jonathan did his postdoctoral training at the MRC Human Immunology Unit at Oxford University, UK. Then later he joined the Cell Death and Inflammation Unit headed by Peter at the VIB Ghent Center for Inflammation Research, where he acts as a principal investigator and associate professor. Jonathan has been interested in the interplay between viruses and the immune system, and actually he has become a star in the biology of intracellular nucleic acid sensors, such as the ZBP1, the actual topic of his talk today. Okay, so his work has published in top journals such as Nature Review Immunology, Journal of Experimental Medicine, Cell Death and Differentiation, and has contributed to the understanding of the molecular basis of autoimmune diseases and development of vaccines. So I finally want to mention that Jonathan received the ARC grant and a prestigious Odysseus Junior grant, among others. And I will cut this short again, Jonathan, sorry, just for the time. Thank you so much.
Thank you so much for this very kind introduction. Can I ask the host to, because I'm unable to share actually.
Oh, okay.
Márcio, coloca
sorry.
o Jonathan como co-host, por favor, para ele compartilhar a tela. Márcio. Obrigada.
Yeah, I think it's okay now. There you go. Share.
Yep.
Think.
Yep.
Yeah.
Just not in the presentation mode yet.
Yeah, it's a bit slow. I have to be very patient.
Okay, now. Great.
Yeah.
Thank you.
Getting there.
Yep.
Pointer. Yeah, there we go. So okay, I would like to talk about how cell death can restrict virus infection. And the main focus will be on ZBP1 or Z-DNA binding protein 1 and necroptosis. So this is a layout of my presentation. So I will first briefly introduce the definition of antiviral cell death, introduce some important concepts in host-virus interaction, because of course antiviral cell death is an interaction between a host cell and a virus. Also introduce nucleic acid immunity, and then talk about ZBP1 as a Z-nucleic acid sensor, and how hopefully, and then I'll follow with some experimental data showing how ZBP1 can restrict virus infection by inducing necroptosis. And if I have some time, I would also like to spend some time on, and this is related to a previous question, in fact, how spontaneous activation of ZBP1 in the absence of virus infection can cause autoinflammation. Okay, so first the definition. So what is antiviral cell death? It can be any form of regulated cell death. So cell death which is regulated by genes, programmed by genes. And this includes apoptosis, necroptosis, and pyroptosis. And what it basically does, it actually is an altruistic way of a cell of saying, look, I'm going to sacrifice myself for the sake of the organism. So it immediately destroys a viral niche of replication. But it also can induce the release of DAMPs, damage-associated molecular patterns, which can then further induce inflammation. And this is particularly important in the context of necroptosis and pyroptosis. And also importantly, it can facilitate the uptake of viral antigens and presentation by antigen-presenting cells. It's also important to mention that antiviral cell death actually is induced before a complete infection cycle is completed. and should not be confused by another form of cell death, which I call proviral cell death. And this, in fact, can occur at the end of an infectious cycle and is important for viral spread. And proviral cell death can be induced accidentally by viral replication that puts so much stress on a cell that the cell bursts, but it can also be programmed by the virus itself. So certain virus proteins can also induce proviral cell death to promote the viral egress from a cell. But I'm not going to talk about that. And very importantly, almost all viruses, especially large DNA viruses, which have a very large genome and which require long replication cycles, they antagonize antiviral cell death. And this makes it indeed a host-virus interaction. So what I also find fascinating, in fact, is when we look at genes or sets of genes that are positively selected across different clades of animals, so across the animal kingdom, we find that here the necroptotic pathway, in fact, in this case in birds, is highly enriched in positively selected genes. So this means that this provides an argument, in fact, that necroptosis has evolved as an antiviral strategy, and other genes, including genes that restrict influenza A infection, herpes virus infection, and cytokine-related. I know these are quite broad terms, but all these genes are immune-related and include the necroptosis pathway. So I think this is a very strong argument, in fact, in favor of necroptosis as an antiviral defense strategy. And before I proceed, I would like to mention this book, actually, which I read after my studies. I wish I had done it before, but it's a book by Richard Dawkins explaining, it's a very clear book, it's very well written, and explains a hypothesis which is called the selfish gene hypothesis. And what he states is, in fact, is that the unit of natural selection is not the individual, as I had assumed before, but it is, in fact, the gene, and that individuals are merely vehicles for genes. And especially in host-virus interactions, I think this is a very important concept to remember. And then the second concept is the hypothesis of the red queen hypothesis, and this was proposed by Lee Van Halen in the 70s. And in fact, it comes from a book from Lewis Carroll, Alice in Wonderland Through the Looking Glass, where the red queen is saying to Alice, it takes all the running you can do to keep in the same place, and if you want to get somewhere, you must run at least twice as fast as that. And this, in fact, is a very important concept in host-virus interactions. And basically what it says is that in order to survive both the host and the virus, and if we take the hypothesis of Richard Dawkins into account, in fact, the genes of both organisms need to constantly evolve. And that host-virus interactions is a dynamic and not a static process. And then the final concept was actually written down in a review published by Jonathan Kagan, an immunologist at Harvard University. And it stresses the importance of imperfection, and Peter also referred to this. But I think it's very important if you think about what actually Jonathan Kagan is writing, is that successful virus infections do not activate pattern recognition receptors and downstream innate immune responses. And this poses a great problem for us. If viruses were to be perfect, we would not exist. And what he says is that infidelities in viral infections can create ligands for pattern recognition activation. For example, and this is shown here on the figure on the left, nucleic acids can be degraded inside the endosomes, and these then, from viruses, these can be sensed by toll-like receptors, for example. Or infidelities in viral replication can lead to an accumulation of aberrant virally derived nucleic acids, which can then be sensed by pattern recognition receptors. And I think this is a very important concept in immunity in general. I would also like to add to that is that imperfect host defense mechanisms are probably responsible for allowing pathogen replication. So if we take all these concepts together and we think about the implications for host-virus interactions, but it may also have important implications for your future field of research, for example, is that host-virus interactions are not static, and then instead, host and viral genes are dynamically co-evolving, and this enables stable coexistence. And this is referred to as the evolutionary arms race, or the red queen hypothesis. Now, a question that I had actually is that assuming that viral genes can evolve much faster, then why are we still alive? You can imagine pathogens, they replicate much faster, their genes are more prone to be to mutation. So why are we still alive? And I think the answer lies within our genetic space, which is much, much larger than those of pathogens. And I think host redundancy, and this is also a concept that Peter alluded to in his talk, the redundancy between the different cell death pathways, for example, this can provide protection against fewer but faster evolving viral anti-host genes. And another important point is that host and viral genes are not perfect, and these imperfections ensure coexistence. So you can imagine if perfection is achieved by one of the two, by either a viral gene or the host gene, this can lead to extinction. And I think there are some examples of animals that became extinct or to the brink of extinction by a pathogen. So the end result would be very sad, in fact, for one of them. So we can take it one step further, and we can think about actually host and viral genes being into a common gene pool. And this may justify each other's existence. So, but this is perhaps taking it a notch too far. So within this framework, we would like to work, and this is how we think about host-virus interactions and between and also on antiviral cell death. So there's another concept that I would like to introduce before we move on to ZBP1, and this is the principle of nucleic acid immunity. And in fact, if we think about viruses, and this is actually an electron micrograph of an HIV virion, and what a virus is in fact, it's just a packet of genetic material. And in the case of HIV, what you find is two strands of single-stranded RNA, which are enveloped by a capsid and a matrix here in purple. And these are proteins, of course. And this is enveloped in the case of HIV because it's a membrane virus by a lipid bilayer. And all these proteins and lipids, they derive from the host cell itself. So it forms a difficult challenge for the immune system to detect viruses. So the way our cells have dealt with that is that they actually sense the difference in biochemistry between host RNA and DNA and between viral RNA and DNA. And we call this, in fact, nucleic acid immunity. So the way a host cell detects an incoming virus is mainly based on the detection of virally derived nucleic acids. So, and this is shown here in this cartoon. So the cell contains different families of nucleic acid sensors, and you may know some of them. For example, the toll-like receptors, which reside in the endosomes, for example, TRR7 and 8, they sense single-stranded RNA in the endosomes. And the endosomes are, in fact, a very important route of infection by the viruses. So they're strategically positioned within the endosome to sense incoming viral RNA. We have other families like the Regai-like receptors, which are double-stranded RNA sensors inside the cytosol. And then there's CGAS-STING, also a very important double-stranded DNA sensor, which actually resides in the nucleus and the cytosol. And what these nucleic acid sensors do is they sense either viral RNA and DNA. Then they induce a transcriptional program leading to the production of type 1 and 3 interferons, which are in fact these cytokines with antiviral activity. And these, in an autocrine and paracrine manner, they bind to the interferon 1 or type 3 interferon receptor. And this induces STAT1 JAK-STAT signaling cascade, leading to the induction of hundreds, if not thousands, of what we call interferon-stimulated genes or ISGs. And it's in fact these ISGs that perform the effector functions that restrict virus infection. And there are several, well, as I mentioned, there are hundreds of these ISGs, but what they in fact do is that some of them can, for example, such as ZBP1, can induce cell death of the infected cells. Others are specialized in host cell shutoff. For example, PKR is a double-stranded RNA sensor, which is a type 1 interferon inducible, and which can then shut down protein translation. And the OAS RNase L system, in fact, is also a sensor of double-stranded RNA. And they can activate RNase L, which is an endonuclease, which in fact cleaves, RNA inside a cell, so resulting in host cell shut-off. So as a summary, so what is nucleic acid immunity? It's recognition of abnormal nucleic acids which are not present or only lowly present in low concentrations in healthy cells by proteins called nucleic acid sensors that activate a signaling pathway to initiate an antiviral immune response. And nucleic acid sensor activation by viral nucleic acids induces the production of type 1 and 3 interferons, so these cytokines with antiviral activity, which act in a para or endocrine manner to induce the expressions of hundreds of ISGs. And one of these ISGs, in fact, is Z-DNA binding protein 1 or ZBP1. And this activation of ZBP1 results in antiviral cell death under the form of apoptosis and necroptosis. And I'll show you a bit later how this occurs. And again, stressing the importance of these pathways, in fact, is viruses, almost all large DNA viruses, in fact, have evolved manners to deal with nucleic acid immunity and with ZBP1 activation. All right, so what is ZBP1? ZBP1, in fact, is part of a family of what we call Z-nucleic acid binding proteins. On this slide, you see that mammals, in fact, express two proteins which are able to interact with Z-nucleic acids. And the interaction with Z-nucleic acids is made through these Z-alpha domains. So this is ZBP1, and this is the domain structure. You can see that ZBP1 has two N-terminal Z-alpha domains, followed by three RIP homotypic interaction motifs. And these are the domains that can induce necroptotic signaling, as Peter has introduced before. Then there's ADAR1, which has two N-terminal Z-alpha domains, followed by three double-stranded RNA binding domains and an editase domain. And if I have some time, I will also show some data on ADAR1, how ADAR1 can act, in fact, as a negative regulator of ZBP1 activation. So, in fact, you can see ZBP1 as an activator of antiviral immunity and ADAR1 as an inhibitor of antiviral immunity. Interestingly, fish express a ortholog of PKR, which is called PKZ, which has two Z-alpha domains and a kinase domain. And also viruses, such as the pox virus, vaccinia virus, has a protein called E3L, which has a Z-alpha domain, through which it can sequester viral Z-nucleic acids away from ZBP1. And there's a fish virus as well, which infect carps, which can also expresses Z-alpha domain containing proteins. So these again are viral inhibitors of Z-nucleic acid mediated immunity. So you may not be familiar with Z-nucleic acids, and in the following slides I'll give some background on what these Z-nucleic acids exactly are. So these include Z-DNA and Z-RNA, and they were in fact discovered in the late 70s by Alexander Rich, a molecular biologist, Alexander Rich. And in fact, the first atomic resolution crystal structure of double-stranded RNA was not in a P conformation, as you would expect from the typical Watson-Crick helix, but it was in fact in the Z conformation. And this was published in the late 70s. And this created a lot of confusion in the field, in fact, as to whether, in fact, the Watson-Crick model was correct. But there was, of course, a lot of experimental evidence that Z-nucleic acids are probably not as common as people thought. Okay, so to understand Z-nucleic acids, we have to look at the topology of double-strand DNA and RNA. And this is what's shown on the slide here. So here on the left, we have a Z-DNA helix and sign. Then in the middle, we have a classical double-stranded RNA helix, which adopts the A conformation. And here we have a classical, this is actually the Watson-Crick double-stranded DNA helix, which we call the B conformation. And the biggest differences between Z-DNA and Z-RNA, which in topology looks very much like Z-DNA, is in fact that Z-DNA is sort of stretched, it's elongated. It has a shorter diameter of 18 angstroms, it has a longer helical rise, and it takes 12 base pairs per turn. And in fact, the biggest difference is that from the 5' to the 3' end, Z-DNA has a left-handed helical turn, while A and B DNA, aRNA and B DNA have a right-handed helical turn, and they're much more compact. So you can see Z-nucleic acids as a sort of left-handed helix, and a sort of elongated form. So it looks completely different from normal double-stranded RNA and DNA. In fact, the name derives from zigzag. So in fact, the backbone has this zigzag structure, and hence the name zDNA and zRNA was given. So what's the reason for this shape? In fact, as opposed to bDNA and RNA, Z-nucleic acids have a dinucleotide repeat unit consisting out of a pyrimidine and a purine, in which the purine is in the syn conformation, so where the glycosidic bond is in the syn conformation, pointing away from the helical axis, and the pyrimidine is in the anti-conformation. And this creates this zigzag shape of Z-nucleic acids. And in fact, it took a long time, in fact, to accept that Z-nucleic acid had a biological function. And the main reason, in fact, is that main reason for that is the fact that the Z-nucleic acids are highly unstable. And the reason for this is that these the sugar phosphate backbone here that you see are in closer proximity together. And these negative charges, the repulsion between these negative charges, destabilizes Z-nucleic acids. So normally, in a test tube, you would find either bDNA or RNA. What people have found later, in fact, is that there are ways to stabilize the Z conformation, and you can do this by increasing the salt concentration, so preventing these negative charges between the phosphates on opposing DNA strands, but also by increasing polyamine concentrations by nucleotide modifications. For example, guanosine methylation can stabilize DNA in the Z conformation. And finally, there are proteins such as ZBP1 and ADAR1 that can bind to Z-nucleic acids and stabilize the Z conformation. So going back to ZBP1, so if you look at ZBP1 in detail, so as I mentioned before, ZBP1 has these two Z-alpha domains. And at the bottom here, you can see the AlphaFold structure of the two Z-alpha domains of ZBP1, Z-alpha1 and Z-alpha2. And these have been crystallized. And what people have realized that two Z-alpha domains can bind in an anti-parallel manner along the helical axis of the Z-nucleic acid. And this is shown here on this crystal structure here. ZBP1, as Peter has mentioned as well, has not one, but up to 3 RIMs, so rib homotypic interaction motifs. And you also find these in other RIM proteins, such as RIBK1 and RIBK3 and TRIF. And these can form beta amyloid-like fibrils. So they can form beta stacks and form a hydrophobic core in between them. And this basically creates these RIBK1, RIBK3 alternating fibrils, which we call the necrosome. And it also has a C-terminal tail here in purple with unknown function. So ZBP1, in fact, became important the last years, and what people have realized that many viruses, including many important human pathogens, can activate ZBP1. And these include both DNA viruses and RNA viruses. And examples are here herpes simplex virus 1 and 2, varicella zoster virus, the causative agent of shingles and chickenpox, but also influenza virus, SARS-CoV-2, Zika virus, and West Nile virus can be detected by ZBP1. So, all right. So how does ZBP1 signal? So, and this part of this is still hypothesis, because some parts of these pathways we still do not understand completely. But what we hypothesize, in fact, is that all these different viruses, DNA and RNA viruses, when they infect cells, this results in the accumulation of zDNA and zRNA, which then binds to the Z-alpha domains of ZBP1, resulting in activation of these RIM domains here, which can then recruit the RIM domain containing kinase RIBK1, which then recruits through the that domain FOD and caspase-8, resulting in apoptosis. And I would like to mention that in the case of ZBP1, but also in the case of TNF receptor, like Peter has explained before me, is that cell death downstream of ZBP1 is a hierarchical process. So when ZBP1 is activated, first cells undergo necroptosis. However, caspase 8, what it does when caspase 8 is active, it can cleave RIPK1 and inactivate it, preventing necroptosis. However, when caspase 8 activity is blocked, ZBP1 can recruit RIPK3, which will down form a necrosome, resulting in the activation of MLKL and resulting in necroptosis. And if you think about it, many viruses, DNA viruses, what they do is they inhibit caspase 8. So many DNA viruses, herpes viruses such as cytomegaloviruses, herpes simplex virus 1 and 2, they express proteins which specifically inhibit caspase 8. And perhaps the most famous example is VFLIP, which is expressed by Kaposi sarcomas herpes virus. So what they do is they prevent ZBP1 dependent apoptosis by blocking caspase 8. However, what these viruses then do is sensitize these cells to ZBP1 dependent necroptosis. And in fact, what Ed Mokarski is referring to this as necroptosis as being an evolutionary trapdoor to caspase 8 dependent apoptosis. And I really like this analogy. So, of course, viruses have found ways to antagonize ZBP1 dependent necroptosis, for example. And I mentioned this protein before, this E3L protein encoded by vaccinia viruses has its own Z-alpha domain, leading to sequestration of Z-nucleic acids, thereby preventing downstream ZBP1 activation. More interestingly, and I'll come back, this is also the system that we use in our experiments, there are viral Rim-containing proteins. So I mentioned these 4 Rim proteins, including ZBP1. What viruses also have Rims? So, for example, MCMV M45 and HSV1 ICP6, in fact, contain at the N-terminus these Rim domains, which can interfere with the interaction between ZBP1 and RIPK1 and RIPK3, and this is how they block downstream necroptosis. Also, there are also other options, for example, proteasome-mediated degradation of RIPK1 and MLKL, and examples of these are UL36 encoded by cytomegalovirus, and the viral inducer of RIPK3 degradation encoded by variola virus, which is the causative agent of pox virus. So to summarize, viruses have, especially these large DNA viruses, which need a lot of replication time. They can be sensed by ZBP1, however, they block both caspase 8 mediated apoptosis and RIPK3 dependent necroptosis. Okay, so how do we study this? The importance of this necroptotic pathway in vivo, and one way to do this, in fact, is to take mice, infect them with a pathogen, and see whether necroptotic pathways, or in this case, ZBP1 is important in virus restriction. So what we did in this particular experiment is we infected wild-type mice, so ZBP1 plus mice, either with a wild-type cytomegalovirus. It's in fact a mouse cytomegalovirus, so it's endemic to mice. And what we know is that wild-type MCMV both inhibits caspase 8 and necroptosis. So if you infect these wild-type mice with this green virus here, and a couple of days later we take out the spleen and see how virus replication occurs, you can see that this virus replicates very efficiently in the spleen of wild-type mice. However, when we infect these mice with a virus which contains a defective Rim-containing antagonist of ZBP1 signaling, and this protein is called M45. So only the Rim domain of this protein is mutated. You can see that no virus is replicating whatsoever in wild-type mice. But then if you repeat the experiment in mice which express a deficient version of the ZBP1 protein. So what we did in particular is we mutated both Z-alpha domains of ZBP1 here, this is shown here, rendering the protein unable to bind to Z-nucleic acids. All of a sudden you can see, of course, the wild-type virus doesn't care whether ZBP1 is there or not because it is able to inhibit it. However, we can restore, or at least partially restore, replication of an M45 Rim mutant virus in these mice in which ZBP1 signaling has been made defective. So this is an example of how we can study the importance of necroptosis and viral antagonism in vivo. So again, how does it work? So what we did to address this question is, in fact, we took immortalized ZBP1 knockout maps and reconstituted them with different versions of ZBP1, in which we reconstituted either with wild-type ZBP1, and what we did is when we infected these with an MCMV virus, you can see that these cells, by increasing the more, you can see that these cells are dying by necroptosis. We know it's necroptosis. We can biochemically probe for that by using an antibody which is specifically directed against the phosphorylated form of MLPL, as seen here. You can see cells expressing wild-type ZBP1. You can see MLPL phosphorylation. But you can also see a reduction in the production of a viral protein, in this case, immediate early protein 1, showing that indeed ZBP1 activation can restrict viral infection. However, when we mutate both cell alpha domains of ZBP1, you can see that cell death is not occurring anymore. And also there's no necroptosis occurring. So this is just to give you an example of how we can study antiviral necroptosis both in vivo and in vitro. All right. So of course this was all done in mice. So we wondered whether this pathway was also active in human cells. And for this we took the approach of infecting cells with a human virus. In this case, herpes simplex virus 1, which is a large DNA virus, a member of the herpes virus family, one of the 9 human herpes viruses. It can be lytic in epithelium, and it also establishes a lifelong latent infection in sensory neurons. And you may know it from reactivation, and it causes these nasty cold sores. In immunocompromised patients, herpes simplex virus 1 can also cause very severe conditions, such as herpes simplex encephalitis, and cause severe eye infections. And it's very prevalent. In fact, most of us by our 40s, 80% of us are infected with herpes simplex virus 1. So it's a very important human pathogen. And to study how this virus can activate ZBP1 is what we did is we took human cells and reconstituted these cells with a GFP tagged version of ZBP1. So these cells express ZBP1, which is present in the cytosol here. You can see this on the left, and the cells are also stained with Huys, which stains the cell nucleus, and with PI. And we added PI, which is propidium iodide, to the medium. And it's, in fact, PI is a cell impermeable dye, which is not able to cross the cell membrane unless the cell membrane is not intact anymore. Then this dye can enter the cells. And it's actually a measure of cell death. So what we did is we infected these cells with, with wild-type herpes simplex virus 1, and like MCMV, HSV1 encodes proteins which block both caspase-8 dependent apoptosis and RIPK3 dependent necroptosis. So, okay, I hope you can see it. After a while, you can see that ZBP1 forms these very nice cytosolic aggregates. However, there's no PI positivity. So this, in fact, shows that ZBP1, after activation, forms aggregates. However, the cells are not dying because the virus is antagonizing ZBP1 dependent cell death. Now, if you repeat the experiment this is an experiment, in fact, analogous to what we did in the mice. We only did it now in vitro in human cell lines when we infect these cells with a strain of HSV1 in which we mutate the RIM protein of herpes simplex virus 1, which can antagonize necroptosis. You can see again that ZBP1 starts to aggregate, but then shortly after aggregation, the cells take up propidium iodide, and we know this is necroptosis. So in fact, what we conclude from this experiment is that ICP6, so this RIM-containing protein, can block ZBP1-dependent necroptosis, and that ZBP1 forms aggregates upon activation, and this precedes necroptosis. So to us, this was a sort of this points to an activation mechanism of ZBP1, and importantly, ICP6 is unable to block ZBP1 aggregation. And to understand the molecular principles of this process a bit better, we looked again at ZBP1, at the domain structure of ZBP1. And as a reminder, ZBP1 has these two Z-alpha domains, which can specifically interact with these Z nucleic acids. And at the C terminus, it has these three rims, which can form rim-mediated beta-amyloidal aggregates. So we thought, okay, these rims are responsible for ZBP1 aggregation. And in fact, to test this, what we did is we mutated, we expressed either wild-type ZBP1 in our cells, either mutate both Z-alpha domains, so preventing Z-nucleic acid binding to ZBP1, or we mutated the rims of ZBP1 individually. So we mutated rim A, B, and C. And then we had versions of ZBP1 only expressing the Z-alpha domains or only the rims. And then I'm not showing any movies again, but when we quantified ZBP1 aggregation, in fact, what we found is that these rims were totally redundant for ZBP1 aggregation. And in fact, Z-alpha domains of ZBP1 that induce these aggregates of ZBP1. And you can see here, this is wild-type ZBP1, which produces around 40 aggregates of ZBP1 per cell. However, when you mutate both Z-alpha domains, this is not the case. And when we mutate these rims individually, there's no effect on aggregation. And interestingly, if you have no rims at all, you can still induce ZBP1 aggregation. So this occurs totally independent of rims. So we were a bit stuck there, but in fact, in the next experiment, what we did is we looked at the stability of these ZBP1 complexes. And what we did here, we took a photo bleaching approach. So what you do, which is called PrEP, fluorescent recovery after photo bleaching. So what you do is you photo bleach using a laser, and this is again doing live cell microscopy. You photo bleach a ZBP1 aggregate, and then you look whether these aggregates, they recover. So whether this answers the question whether ZBP1 molecules are moving in and out of these aggregates. As you can see, if you photo bleach a wild-type aggregate, there's no recovery. And this tells you that these ZBP1 aggregates are extremely stable. However, when you photo bleach an aggregate which contains a mutation in the first rim of ZBP1, you see some recovery, showing that these ZBP1 aggregates are not as stable as wild-type aggregates. And in fact, it's even more pronounced when you look at a ZBP1 molecule which only has the Z-alpha domains. So you photo bleach these, and then you wait 3 minutes later, and you can see that there's some recovery while there's none with wild-type ZBP1. And this is quantified here on the right. So what it means is that these rims of ZBP1, that in fact the Z-alpha domains of ZBP1, they detect Z-nucleic acids. This leads to aggregation of ZBP1. But in order to form stable aggregates, you need rims of ZBP1. And we hypothesized that these rims are in fact beta-amyloidal structures. So you can test this using an approach which is also used in Alzheimer research, in fact, and this approach is called semi-detergent agarose SDS agarose gel electrophoresis. Okay, so what you do is you instead of running your proteins on an acrylamide gel, you run them over an agarose gel with larger pores, so you can separate larger protein complexes. And you do this in the presence of 2% SDS. And normally, normal protein complexes will fall apart in the presence of such a high concentration of detergent. But in the case of amyloidal complexes, these are highly stable, and these are resistant to 2% SDS. And this is exactly what we see for ZBP1. So when we run protein lysates expressing wild-type ZBP1, you can see that ZBP1 forms these higher order SDS resistance oligomers. And we hypothesized that these are these beta-amyloidal complexes which are formed through rim-rim interactions through ZBP1. And you don't see this when you mutate both Z-alpha domains as expected, but you also don't see it when you mutate the first rim of ZBP1, but not the second and third rim of ZBP1. So, of course, we did a lot more of these experiments, but this led us to a sort of ZBP1 activation model in which we think that under conditions of stress, for example, when a virus infects a cell, this results in the formation of zRNA and zDNA. This is sensed by the Z-alpha domains of ZBP1, bringing 2 or more ZBP1 molecules in close proximity. And this results in proximity-induced oligomerization of these RIMs. And we think these are actually the beta amyloids that you find between RIPK1 and RIPK3 as well. And this forms a stable signaling complex, then resulting in the recruitment of RIPK1 and RIPK3, resulting in downstream signaling and necroptosis. So I'm not sure if I have some more time, or should I stop here?
We have, a couple of like 5 or 10 more minutes.
Okay. Yeah. Okay. So then I explain. This relates to how you can imagine that nucleic acid immunity is a very powerful concept because you can detect in a very efficient way a virus infection. But on the other hand, if you think about it, it's also very dangerous because our cells are packed with RNA and DNA molecules. So again, there must be a tight regulation in preventing sensing of our own RNA and DNA molecules. And this is exactly what I will talk about in these final slides. This work relates to ADA1, which is next to ZBP1, the second Z-nucleic acid binding protein. And what ADA1 does, in fact, is ADA1 stands for adenosine deaminase acting on double-stranded RNA. What it does, it binds to endogenous double-stranded RNA, and it removes this amine group here from an adenosine nucleotide and converts it into an inosine, specifically within double-stranded RNA. And this results in the destabilization of double-stranded RNA molecules, so unfolding of double-stranded RNA. And this is very important to prevent spontaneous detection of double-stranded RNA by nucleic acid sensors such as MDA5 and PKR, and this can then result in a spontaneous type 1 interferon response and in spontaneous pathology. So what we in fact wondered is whether ADA1 is also a negative regulator of ZBP1, because it has this N-terminal Z-alpha domain. And we think this is also important for human disease, because in fact what scientists in the 2000s have found is that there is a disease called Aicardi-Goutieres syndrome, which is caused by mutations in the gene encoding for ADA1. And what's particular about this disease is that in 50% of the patients, you'll find a compound heterozygous conformation of the ADA1 mutations, in which a first allele hits this Z-alpha domain here of ADA1, rendering the Z-alpha domain of ADA1 dysfunctional. And it's always combined with a second allele, which is a null for ADA1. So what these patients express is basically 50% of ADA1 protein, which is unable to bind to Z-nucleic acids. And what these AGS or Aicardi-Goutieres syndrome patients, they develop severe brain encephalopathy, so brain inflammation, which is accompanied by physical and mental retardation. This comes likely from calcification of the brain due to the inflammatory response. And what's very typical about these patients is that they develop a spontaneous type 1 interferon signature. And in fact, Aicardi-Goutieres syndrome is part of a bigger family of diseases, which we call the type 1 interferonopathies. And this is a group of diseases which is associated with spontaneous type 1 interferon signaling. Okay, so to understand this process a bit better, what we did is we developed a mouse model mimicking the allelic combination, which is also found in patients. So for this, what we did is we generated ADA1, which we call hemizygous ADA1 Z-alpha minus mice. So what these mice are, so they combine a null allele of ADA1 with an allele in which just the Z-alpha domain of ADA1 has been mutated. And what you can see is that these mice, they die right after birth. In fact, all mice die 1 day after birth. And this is accompanied by the spontaneous production of type 1 interferons. And this is in fact measured by measuring CXCL10 in the serum, which is a surrogate for type 1 interferons. So what's interesting is that if you have a combination in which both Z-alpha domains of ADA1 are mutated, these mice are perfectly fine. they have only a minor spontaneous type 1 interferon response, but it's not pathogenic. And if you have, for example, a heterozygous condition in which you combine a null allele of ADA1 with a wild type allele, these mice are also perfectly fine. Only if you on top of that mutate this set alpha domain of ADA1, all of a sudden this is a lethal combination, resulting in a spontaneous type 1 interferon response. So in fact, to answer the question whether ADA1 is a negative regulator of ZBP1 and may contribute to this disease, Aicardi-Gutierrez syndrome, what we did is, again, we performed a genetic process. So what we did is we crossed these ADA1s at alpha minus mice onto a ZBP1 deficient background. And again, we use these mice, maybe you remember from our work with the virus, we use these mice which express a dysfunctional ZBP1 molecule which is unable to bind to Z-nucleic acids. And you can see that again here, these are the ADA1s at alpha minus mice. They die right after birth. However, when we put these mice here indicated by the blue line in a ZBP1 deficient background, 50% of the mice survive till 18 weeks post-infection. So this tells us, in fact, that ADA1 is a negative regulator of ZBP1 and has relevance for this disease model. And also importantly, that sensing of endogenous Z-nucleic acids by ZBP1 can induce autoinflammation. So we talked about the good side, whether that ZBP1 can protect us against virus infection. In fact, there's also a downside when we lose one of these negative regulators. ZBP1 can also induce autoinflammatory disease. I would also like to mention that the rescue is not complete. So there are probably other sensors, nucleic acid sensors, that are regulated by ADA1 to prevent spontaneous disease development. And this brings me, in fact, to the general conclusions that necroptosis restricts virus replication and has evolved as a means to deal with virus infection. That ZBP1, through recognition of Z-nucleic acids, can induce antiviral cell death. That, of course, viruses have evolved ways to deal with ZBP1-mediated cell death. That blockade of caspase-8 sensitizes ZBP1-dependent necroptosis, and this is this evolutionary trapdoor that Admiral Karski proposed, and that ZBP1 forms amyloidal signaling complexes to induce necroptosis, and that, importantly, the downside is that spontaneous ZBP1 activation by endogenous Z-nucleic acids can induce autoinflammation. And this is my final slide. Again, I have to thank Peter Vandenabeele for taking me in his lab, actually a couple of years ago, for putting his trust in me, and our collaborators. This is the team, and this is in fact our newest recruit, Marlon Fortes. He actually obtained his degree from USP. So I'm very excited to have Marlon here. I think he's trained very well at your university, and we're excited to do continue our research on ADA1 together with Marlon. All right, that's it for me. Yeah.
Thank you so much, Jonathan. Was awesome. I have myself, a few questions, but I will leave because of the time restraint to the students, of course. But here is another invitation. I host a weekly immunology seminar along with the Federal University of Rio de Janeiro. So it's a larger crowd of immunologists. So these are Mondays at noon in Brazil. So, if you don't mind, amplify your talk, in the second semester, we keep in touch and we get another
Sure, yeah, that sounds really great. Thanks. Yeah.
Okay, so let's start upside down with the people that already asked questions in the chat. So Leticia, and then, I see Caro first. So Caro, you can start. Turn on your mic and ask. Thank you.
Hello, everyone. Can you hear me?
Yes.
Yeah.
Okay, thank you, Professor Jonathan and Professor Peter for the good presentation. So I happen to be working on microRNAs, and in my literature, I've seen that we have quite a number of microRNAs that have been mentioned on being involvement in cell death. Particularly, we have some microRNAs such as the microRNA 145. And yesterday, Professor Lockshon mentioned that previously, when they were doing some gene analysis, they realized that they have some short RNA that they saw, but they didn't do further analysis to see if they were microRNA or what kind of short RNAs that were there. So in the last 2 decades, there's so much research going on microRNA, particularly on cancer and also other diseases. So my question is, have you, in your research, being that like Peter, his background is on molecular biology, really done, identified some of these microRNAs, and do you think that this microRNA can be used as some checkpoints to maybe block some of these pathways?
Yeah, I will briefly answer. So we did not really study microRNAs in the TNF signaling pathway, but there is indeed, as you also mentioned, there is quite some literature that is interfering with inflammation induction by TNF by microRNAs. But maybe Jonathan can add something to that in the case of ZBP1.
Yeah, it was not necessarily on microRNAs, but yeah, if you think about a lot of these proteins involved in cell death, they have splice variants, and maybe on a similar level, this is controlled by transcription. And what is really important is the stoichiometry between cell death proteins. So high levels of RIPK1 will also sort of skew a cell to necroptosis. And the same is true for different splice variants. For example, splice variants of ZBP1 exist both in humans and mice, and they can act as a negative regulator of ZBP1-dependent necroptosis, for example.
Yeah.
It's not entirely an answer to your question, but
well, it's not directly microRNAs, and so in the case of caspase-8, you also have several splice variants, and indeed, it's a domain that is not studied enough, I think. That there are splice variants of caspase-8 which are inactive, catalytically inactive. And you can imagine that those would also be conditions that sensitize necroptosis. The same if you would have targeting of caspase-8 by microRNAs, which also has been described.
All right, Leticia.
Okay, hello. My question first, thank you for the presentation to Jonathan and Peter. I love what I learned. So my question to Jonathan is if you ever heard or worked with any genetic variant of ZBP1, and how did she behave or bring any susceptibility to cell death?
Yeah, that's a good question. There's none described so far. That's the short answer. I think if we find one, I think it will be related, that's my guess, educated guess, either to herpes simplex encephalitis or West Nile virus infection, because a lot of genetic studies are being done with those particular pathogens, especially since they affect young children. So they point to a monogenic disease. And what recently there has been a paper describing a mutation in RIPK3 predisposing to herpes simplex encephalitis. So I'm sort of expecting, maybe in a couple of years, that such patients will pop up. But so far, no loss of function or gain of function mutations have been identified in ZBP1 so far.
And also in the case of Zika virus, it could be also good cases, especially we are in Brazil and Zika virus was very important there.
Yeah.
Okay, so
My question to, sorry, can I question to Peter?
Yeah.
Okay, my question to Peter is, thinking about the necrosis that occurs in type 2 diabetes mellitus, how would MLKL KL or RIPK1 blockade be interesting?
Yeah. I think it's for sure any disease where you have reduced blood flow and that is causing ischemic conditions. Those are conditions that are good candidate for, let's say, stress that will lead to RIPK1 activation. As I also explained, or tried to explain, is that probably in disease conditions, you will have a mixed response in cells. So there will be apoptosis, there will be necroptosis, there will be probably also, like in ischemic conditions, also ferroptosis. So I think that targeting, of course, RIPK1 is good because it's upstream both to apoptosis and necroptosis. If you look more at the level of MLKL, that is at the more downstream point. The question is, will that be sufficient? But at least targeting MLKL, we are looking now also in screenings for MLKL inhibitors. And the whole idea is indeed that in those conditions, you would reduce the final explosion of the cells, or let's say that you would induce less damp release, and that would improve the whole situation. But I think any condition like indeed type 2 diabetes, but also organ transplantation, degenerative diseases, there you can expect probably contributions of necroptotic cell death.
Awesome. Thank you, professors.
Yeah.
So Letícia Serafim.
Hi, can you hear me?
Yes.
Hi, professors. Thank you for the lecture. It was amazing. I actually never heard of ZDNA, so it was like a really new thing for me. So I don't know if I'm gonna ask something that doesn't make sense, but I did some Google and I saw an article that they found out antibodies anti-ZDNA in people that have lupus. Does it have any connection, or actually was just some binding that wasn't supposed to happen?
It's a very good point. Normally it's in my presentation, actually, this this paper. You're absolutely right. So what's common in lupus is these anti-nuclear antibodies. So lupus patients have antibodies against bDNA as well, and aRNA as well, but also against zRNA and zDNA. So for us, that was quite interesting from a sort of fundamental perspective that these structures would exist in nature. Then the question, of course, is whether it's sensed by ZBP1 and this contributes to disease. That we don't know. I'm personally, as a fundamental researcher, staying clear from very complex diseases like lupus, and focus on monogenic disease to learn like biology. And then I would also like to add these antibodies. In fact, we use them for so we can stain cells for Z-nucleic acids using these anti-zDNA antibodies that you mentioned. So it's actually a useful tool for us.
Cool.
That's a very good question. Thank you.
Great. Bruno. Bruno?
Turn on your mic, Bruno.
Okay, Bruno, while you are trying, let's hear from Barbara. No, don't hear as well. Oh, now I think. Yeah, yes.
Yes. Oh, thank you for your talk. It's great, very, very great. I like very much. So I think would induce ZBP1 in a viral infection be interesting for the death of infected cells and for the recruitment of CD8 T cells to the infection site. And could this be used as a therapeutic tool against viral infections, or no makes sense? I don't
Yeah,
know.
no, it makes a lot of sense. Um, the relationship between ZBP1 and adaptive immunity hasn't been, I don't think it's that clear cut, because there's quite some redundancy. I think it's more an innate immune response, but actually, people have looked in anti-cancer immunity, which also heavily relies, anti-tumor immunity, which heavily relies on CDC1, CD8 T cell responses. And people are actually ADAR1 inhibitory therapies are being used now in the clinic. Yeah, so there are inhibitors that are being developed to treat tumors with ADAR1 inhibitors to activate a double-stranded RNA response. And my prediction would be is that ZBP1 is also among those sensors that is activated. But if you remember, there are other double-stranded RNA sensors like MDA5, PKR, which also sense double-stranded RNA. I'm not sure if that answers your question. But like this 1-1 relationship between ZBP1 and CD8 T cells is a difficult one because of the redundancy, I think, in the system. Yeah.
And the RNA vaccines, there was one of the questions also about RNA vaccines and that form.
Yeah, we haven't tested those yet in our mice. That's a good idea. Yeah.
Okay, so Bruno's microphone is not working, so I'm gonna ask his question. What is the role of ZBP1 in the context of different viral infections? Influenza, SARS-CoV-2, Zika, dengue, all this.
Yeah, most of the studies, of course, have been done in mice, if you refer to in vivo infections. It can go both ways, in fact. So there are studies showing that it's antiviral, clearly antiviral, so the mechanism that I proposed, but it can also contribute to immunopathology. In the case of SARS-CoV-2, which leads to a hyperactive inflammatory response, ZBP1 can contribute to that. That's work from Kaniganti has shown this. So in fact, ZBP1 knockouts are better protected against virus infection. Well, no, that's wrong. They're better protected against virus-induced immunopathology. They're not better protected against virus infection. So they restrict the virus first, but the immunopathology, yeah, it's driven by ZBP1. So it's a bit this balance. See the same with influenza. There are studies showing that ZBP1 knockouts do worse. Other studies show that they do better because you don't have this secondary autoinflammatory reaction.
Okay, Guilherme, I think the last one. I saw that we have other questions and Bolaji raised his hand, but I think we have time only for Guilherme's question, okay?
So hello, first of all, thanks for the presentations. I would like to know if there is any difference between the cell death associated to a pro-viral response and to an antiviral response.
In terms of immunological outcome?
Yeah, in terms of pathways or even type, mechanisms.
I don't think it has been studied. I would guess a pro-viral cell death would be, favoring the virus actually. Because you enter a phase where it's already too late, I think, where the virus is replicated and everything that comes,
So would you think that pro-viral cell death would more involve the activation of pore-forming mechanisms so that you have a rapid release of viral particles.
Yeah, there's a viral MLK molecule. Like you sent me that paper, Peter.
Yeah,
It's very
I know.
interesting. Yeah, there's as norovirus, so the cruise ship virus actually that causes vomiting and this diarrhea, very infectious, has an MLK-like protein, which induces cell death. With viral egress. I can imagine it will be highly inflammatory as well, but probably it's already too late for the patient now.
Yeah, it's a bit like the cytolytic virus. They need to be released to be infected. So, but whether this would be like a controlled response using host proteins or not. All right, guys. I would like to thank you so much.
Thank you so much.
It's a pity that you don't have more time. The next speaker just arrived in the room. Dario Zamboni. Hello, welcome, Dario.
Hello, how are you guys?
Okay. We will make a break in a YouTube and here for 10 minutes so people can.
5 minutes.
We can give 10 minutes so we can rearrange things. Okay. So, and then I know that Dario not gonna be using his 2 hours, full 2 hours, so we can use a little bit. Okay. Yeah. Thanks a lot, Jonathan. Okay, I will get in touch with you again for another specific talk for immunology.
Thank you so much. I felt really
Peter
welcome.
as well. You're always invited for all these courses. You're really great. And thank you for your time. Hope to see you soon.
Yeah, okay? Pleasure. Thank you so much. Bye.
And by the way, I didn't say out loud in a live, but I love your shirt, Peter. you look great in a Brazilian Hawaiian style.
Yeah. Well, at least when I couldn't come to Sao Paulo, at least I'm partially in the atmosphere.
In your mind and food. Exactly. All right, guys.
Okay,
thanks so much.
Thank you.